and I get the, I assume you know everything. Yes, dear? After the test is complete and the due date has passed, you may go in and review. Okay, but not until the due date's passed. Correct. Friday at 11:55 p.m. Yeah, I'm not going to stay up that late. So you can check it out on Saturday. No. But what could you do? No. Or maybe you could take some notes that says, oh yeah, I don't know what an electron is. Maybe I should study that. Study electrons. Or, oh yeah, I have no idea what an energy level is. Or a valence electron. Maybe I should study that. Write that down. Yeah? Why do you need the questions, those row questions? You need to know the information. Right, so now you can take notes on what you got wrong, so you know you need to go study that. But that you can do, right? Okay, so we good? All right. So chapter 3. What's chapter 3? You notice we're kind of getting bigger as we go. So we started with chapter 2, which was what? The smallest building blocks of matter, atoms, electrons, all that stuff. Now we're moving up to what? And what is a cell? The smallest living unit, right? So now we're into life. This thing is made of atoms. Is it alive? No. It's made of molecules. Is it alive? No. But those molecules come together to create cells. And this is where we hit the living portion of thing. Now cells. Cells are like little factories. Self-contained little factories. They are contained by something called the plasma membrane, and they have work to do. The work that they do is going to do what for you? Everything. Help maintain you as an organism. So some of my cells are going to protect me and cover me. And some of my cells are going to help me digest my food. And some of my cells are going to help take that food and dole it out to everybody. You see where I'm going? And each cell has a job to do to help maintain you as an organism. So when we look at cells in the human body, they are very diverse and different, depending on what job they have to do. So if you look at this figure on page 62 in your textbook, you'll see how different cells can look. In this chapter, we'll discuss categories of cells and those functions. So we have cells that connect things to one another. We'll talk about epithelial cells. We'll talk about connective tissue and the cells that are important there called fibroblasts. And what are those red things up there? Erythrocytes, which is a fancy name for what? What the heck does that connect? Yeah, what do red blood cells do for a living? They carry respiratory gases, oxygen and carbon dioxide. So how does that connect all the other cells in my body? What do my cells need in order to make the energy they need to do the work that they need? Oxygen. See the connection? So that's going to be my delivery system. Where does the oxygen come in? Which system? Respiratory system. And where does it need to go? Everywhere. So the circulatory system is going to help deliver. Cells that move parts. Why do we need to move? Yeah, we should go get food, right? To come to class, to survive in my what? to survive in my environment. So skeletal muscle, I bet you're saying, why isn't bone up there? Do bones move us? No. Muscles move us. Bones just happen to be the sturdy point in which the muscles pull on to move us. Yes? We'll talk about that when we talk bone. Storing stuff, very important. Fat cells. Fighting disease. Macrophages. We'll talk about phagocytosis in this chapter. Nerve cells, what are those for? I just, as I'm sitting here saying, what are those for, I did this. Because I wanted to put my foot up on my stool. How the heck did that happen? Yeah, nerve cells. My brain, where I said, geez, I'd like to put my foot up, that would be a little bit more comfortable, then sent message where? Yeah, out my spinal cord to my peripheral nervous system, where it innervated muscle groups for me to lift my leg. We'll learn that when we learn the nervous system and the muscular system, right? So that's how I communicate stuff throughout the body, and that's the nervous system. Nervous cells are very different than all of those other guys. And then, of course, in order for us to survive as a species, what do we have to do? We have to reproduce. So reproductive cells are way different than all those other guys, because as far as their map goes, they only have half a map. Why do they only have half a map? And what is she talking about, maps? What's the map I'm talking about? Genetic material. And two half maps in the reproductive cells are going to come together to make a whole brand new map. That's a whole brand new what? Whole brand new organism. That genetic material is housed inside what organelle? What the heck is an organelle? Well, this is a typical cell. Not all the cells, as you know, because we just went through all the pictures, look like this. But most, and I'm going to say most because there's an exception to the rule right on that page we just went by, most cells have all the same parts. What we're going to see different in one cell or another is maybe we have more of some of these parts or less of some of these parts. But that big part up there, called the nucleus, is one of the very important parts in your cells. And what does the nucleus do for us? That's where the map is. The map is not only going to make you who you are, but it's also going to be directions for your cells to do what they need to do to make you who you are. There's one exception to the rule on that previous page. Does anybody know who it is? Doesn't have a nucleus. Nope, they have a nucleus. You know where it is? Guess where the nucleus is? Right there. Guess what this is? Yeah, it's triglycerides. So it's a big, huge sac. So all the organelles in that cell are all kind of squished to the sides. And that's a big pocket of triglycerides, stored lipids. So did anybody say it yet? Red blood cells. Exception to the rule. They don't have a nucleus. Mature red blood cells don't have a nucleus. When they're born in the bone marrow, and we'll talk about this when we talk blood, second semester. We'll talk a little bit about it when we talk bone. They're born in the red bone marrow. And then they're released into the circulatory system when they're mature. When they're born, they have a nucleus. They spit it out before they go into the circulatory system. Can that cell reproduce? No, because you need the map to reproduce. So this generalized cell you're going to see again. Where are you going to see this again? On your first lab practical exam. Yes? So you need to be able to recognize and label all of the parts. I'm telling you this now. Why? So when can you start studying this? Yesterday, right? What are you studying as we speak? And what else? Oh, remember the little anatomical guy? Because you're studying that now, aren't you? Of course you are. You see where I'm going with how this works? This is your homework. You have to study, you have to learn, you have to memorize. Unfortunately, that's what anatomy is. Physiology, you can get away with a little bit more. But there's no getting around it. You have to recognize it, you have to memorize it, you have to be able to label it. So if you have a hard time with that, I'd highly suggest you start now. Yes? On your lecture test, you're going to have to tell me or understand what all the parts do. Yes? So that's how we break up the anatomy and physiology. A lot of the anatomy part you're going to be tested on in lab. The physiology, how it works, that's what most of your test is going to be in lecture. So the first thing, and we'll go through this guy in a minute, the first thing we're going to talk about of this generalized cell is the wrapping around the outside. So this is a self-contained single cell. And a whole bunch of these will come together to make what? Tissues. Guess what chapter 4 is? Tissues. So today we're talking about each individual unit. The first thing the textbook talks about is the wrapping. Now it's not a solid wrapping, is it? It's called a semi-permeable membrane. And sometimes you will see it referred to as the fluid mosaic. Is this a solid thing? No. It's made of primarily some of the guys we learned about in the chapter before from the lipid family, primarily made of something called phospholipids. And that's, when you look at the diagram, it looks like little clothespins. See them? There's two layers. So we refer to it as a what? It's a bilayer, a phospholipid bilayer. Now when we talk about phospholipids, remember we mentioned last chapter that parts of them are hydrophobic and parts of them are hydrophilic. What the heck did that mean? Some of them, one part of them, what was the part of them that was afraid of the water? Yeah, but besides hydrophobic? What's it called? There's two of them. The molecule. What are fats made of? Fatty acids, very good. Glycerol and fatty acids, right? And a phospholipid has how many fatty acids? 2. And what part of the molecule is hydrophobic? The fatty acids, very good. And they kind of repel water and hang out close to each other. So where do you think the hydrophilic end of the molecule is? Yeah, this little ball part, the phosphate part. Because what's all here? It's nice up there that they make it blue for us, but what is it? It's water. It's some kind of fluid medium. Inside the cell, fluid. It's called cytoplasm, or cytosol if I'm just talking about the fluid. Bunch of water. On the outside, bunch of water. So the hydrophilic ends point towards the water environments, and the hydrophobic ends point towards each other. They form a phospholipid bilayer. Is that all? That's it? That's the plasma membrane. That's going to be easy to study. There's more, yes? There's more on the diagram, yes? A whole bunch more on the diagram. Because the plasma membrane, or what else did I call it? The fluid mosaic model has a whole bunch of other stuff that are going to allow, we also sometimes refer to it as a semi-permeable membrane. What the heck does that mean? Certain things can go in and out. So all of these other guys are going to help with the certain things in and out part of the plasma membrane. So these big purple things, and we'll see them throughout the whole textbook, sandwiched in that phospholipid bilayer are certain proteins. Now some of the proteins go all the way through the plasma membrane. What do we call those guys? They're called integral proteins. And then some of them kind of either hang out here or hang out on the outer edge. What do we call them? Peripheral proteins. All over the place, different jobs. We'll talk about them as we go. Now, some of the lipids, the phospholipids, have these little green whatsases hanging on them. Now, what do you think the green whatsases are? They look familiar. You say to yourself, I have seen that before. Where have you seen that before? Last chapter. We're getting warmer. They're carbohydrates. Now, carbohydrates stuck to some of these other structures, like lipids, or some of these big purple blobbies, have a very specific job as well. So we see this guy here stuck to a lipid molecule, and we call it a what? Glycolipid. What do you think we call one of those jobbies stuck to a protein? Glycoprotein. And again, those carbohydrate molecules have a very specific function that will help the cell do what it needs to do for a living. They could be signs or markers, not only for certain functions to take place, but they also might be recognition signs. When we talk about the lymphatic system and immune response, we're going to talk about these guys again. Because they're sort of like street signs that say, hey, here I am, this is who I am, to all the rest of the cells in your body. So they have a wide number of jobs that they do. So we have phospholipids, we have glycolipids, we have glycoproteins. What's the yellow things all kind of squished in there? Yeah, this, remember we talked about cholesterol, big steroid, lipid family. Remember I said it was really important, it was part of your structure for every single plasma membrane of every single cell in your body? Well, you see it now, because it's part of the membrane structure. So where do I get cholesterol from? Like the foods that I eat. Spinach? Is there any cholesterol in spinach? Animal. You, animals. Animals have cholesterol as part of their cell structure. You name me one vegetable I can get cholesterol from. No. I didn't ask you whether you could get fats from vegetables. That wasn't the question. But there's no cholesterol in plants. There's lipids in plants. There's saturated and unsaturated lipids in plants, but there's no cholesterol. Yes? That's an animal thing to use to make our plasma membranes. So the different membrane proteins are going to perform different functions for us. Integral proteins versus peripheral proteins. And integral proteins, a lot of times we'll see, act as like little bridges to allow things to come into and out of cells. Certain substances control these little bridges, and they sometimes act in different ways. We're going to talk about them in this chapter. Sometimes, as you're reading, they will be referred to as ATP ACEs. ATP is what? That energy molecule, ACE, A-S-E, at the end of a word. Enzyme. Some of these guys are going to help us produce and use energy to perform a different function. Okay? So when you read that or see that word, don't get confused. That's basically what they're referring to, that function. They talk a little bit about something called the lipid raft. About 20% of our outer membrane surface contains a lipid raft. And this has to do with different concentrations and platforms and receptors, those glycolipid, glycoprotein guys I was talking about on the surface of cells. So, membrane's not just phospholipid. It's phospholipid, cholesterol, glycolipid, glycoprotein, integral proteins, peripheral proteins. Kind of complex. Should you understand what all of those are? Yes. And you should understand their function. So, let's talk a little bit about some of those proteins. There's a whole bunch of different types of proteins on the surface or within the surface, within the membrane of the plasma membrane. The first one they talk about in the textbook is something called a transport protein. Again, acts like a little bridge. Certain ones will allow things to move in and out. When we talk about water and osmosis, the movement of water from a high concentration to a low concentration, we'll talk about one of these little transport guys called an aquaporin. What do you think that means? Yeah, allows water to do what? Kind of like a pore, allows water to move in and out of the cell. As we discuss cells and cellular function, we'll see how important water concentration is to proper cellular function. Some of these act as receptors. When things come in contact with them, they're going to trigger a chain reaction within the cell and cause the cell to do something different. So in your textbook, we see receptors for signal transduction. Don't be scared by that term. Basically, it just means a chain reaction of chemistry. Starting a signal. If I was to line up dominoes on my desk here, that receptor would be like me knocking the first domino down. Its change is going to then cause all the other dominoes to move, or a chemical reaction chain to happen within the cells. Some of them are basically for attachment in a couple of different ways. One of the things we're going to learn is in order to create tissues, what do I have to have? A whole bunch of cells. And how do I hold the cells together with some of these proteins? So some of them are to attach to each other. And we see them described as attachment to the cytoskeleton and extracellular matrix. What's extracellular matrix? Extracellular. Outside the cell, exactly. So it will allow a little structure outside the cell to hold on to somebody else to form a tissue. There's different ones, and they talk about it in the discussion of cellular junctions. We'll talk about that in a minute. Some of these proteins are going to act as enzymes. And what's an enzyme doing for me? Remember the ASE thing we just went over? What's an enzyme do for me? Chemistry. It's a catalyst. What's that mean? It's going to help speed up chemical reactions and lower the amount of activation energy necessary to do it. Everybody understood what I just said, right? Because in order to drive reactions, and let's call them metabolism. That word metabolism is a general term for all of the chemistry that goes on in our bodies. So we need energy to drive reactions of metabolism. So we're constantly having to make it. If I can decrease the amount of energy I use, that's a good thing, right? Say yes, that's a good thing. See, it took you energy to do that. Maybe I shouldn't make you shake your head. I will be the enzyme. So, enzyme activity is also one of the jobs of some of these proteins on the membrane. Joining cells together, intercellular joining. And then, as I mentioned before, sort of the street sign. Now, why do I need street signs on my cells? Why? There's a lot of cells in my body, right? And there's a group of cells that are constantly going to monitor who's who in your body. Do you know what system that is? The immune system, right? And those cells are kind of like the security guard that walks around a building, constantly going into each of the rooms, checking to see that everything is okay. Well, how do those cells know that your cells are your cells? How do those cells know that a bacteria isn't you? Cell recognition proteins. Okay? Your DNA is going to determine what kind you have. So indirectly, yes, DNA. But your DNA is going to direct the type of proteins you have on the surface of your cells. And then your cells, the cells of your immune system, actually have to go to school and learn who's who. We'll talk about that when we talk about the thymus. And we talk about a group of cells called T cells when we discuss an immune response. Those cells know by learning which proteins, cell recognition proteins, are yours. Every single living cell has them. Mine are different than yours, are different than bacteria's, are different than other organisms. So that's how my body protects me, constant surveillance. Everybody with me? So when we talk about cells sticking together, the other thing we're going to learn is cells in groups of tissue also not only have their own little jobs to do, but they sort of work as a group and have to be able to communicate with each other in order to perform different functions. Clicker down. I like when the batteries pop out and they roll all the way down. That's awesome. Okay, so cells have to be able to communicate with each other quickly as well. So not only do they have to hold on tight to each other, but they also have to be able to communicate with each other. So when we discuss different cell junctions, things that hold cell together, we describe some very different things. The first one the book talks about is something called a tight junction. And a tight junction is kind of like a Ziploc bag. And you know it's trouble when you think it's sealed, but it's not. Does that ever happen to you? Yeah, makes a terrible mess. But when it's sealed nice, it holds things together nice, the two ends of the bag, so everything doesn't spill out. So tight junctions kind of remind me of Ziploc bags. They're impermeable junctions, so water can't flow through them or things can't flow through them that hold things together. Now that's kind of important for the cells that make up your bladder, yes? Kind of want them to hold on to each other good and tight. How come? Because if they didn't, all your urine would spill out into your pelvic region. Not good, yes? We want to hold on to it and then get rid of it out of our bodies, because what is it? It's waste. You see where I'm going? The other one they discuss is something called a desmosome, and that kind of reminds me of Velcro. It's a nice, tight fit. If you ever get really bored someday, go look at Velcro, and you'll understand what I mean. It's got like a little hooky end and a little fuzzy end, and the little hooks go, you know. I guess I've been bored in my life. So that's going to help anchor things tight, bind one cell to another, and we're going to, with the help of some very strong proteins, be able to hold one cell to another. And the last one they describe is something called a gap junction. And a gap junction is sort of like taking a straw and poking it through two plasma membranes. So it goes from one side to the other. Now, what's the point of that? She said, we're trying to keep the cells separate. We're trying to stick them together. We don't want anything to leak out. And then we go and stick a straw through both plasma membranes. Yeah. When you sit here, as you sit here, there's a whole bunch of stuff going on. You're not just sitting around doing nothing. You're breathing, right? Your heart is beating, which is kind of good. And that does that 24 hours a day, 7 days a week, for your entire life. In order for the muscle, your heart, to beat, it needs chemical stimulus. It gets some of it from the nervous system, but it actually gets it from communicating with each other, that group of cells in your heart muscle. When we talk about the cardiovascular system, second semester, we'll talk about a group of cells called autorhythmic cells. And they kind of keep this chemistry flowing with the help of those gap junctions through your muscle tissue in your heart, so that your heart can, through a wave of chemical change, contract and relax and contract and relax. And what's it do for a living, your heart? Yeah, pumps blood. That's important because blood is your delivery system. So I constantly have to keep it on the move throughout all of your different organ systems. So understand the differences between tight junctions, desmosomes, and gap junctions. Now I said your membrane was a semi-permeable membrane. So before we talk about the part about the membrane part being semi-permeable, we want to understand that you understand how molecules move through space. So we're going to talk a little bit about transport through a membrane by defining things like diffusion. Now, what is diffusion? We talked a little bit about this when I used that analogy of you getting all squished in this room, and we talked about equilibrium reactions. Remember that? Because molecules are always what? They're always moving. They're always in motion. And where do they want to go? They want their own space. They want to go from high concentration to low concentration. That's the definition of diffusion. And we can see in this little beaker demonstration, if I put this little colored dye thing in there with certain molecules, it's going to, just like my sodium chloride crystal in the other example, because the molecules are in motion, somebody's going to break off. And water, because of its wonderful little polarity, yes, its little attraction that it has, that slightly negative and slightly positive thing going for it, is going to help distribute those dye molecules, eventually evenly distribute them through the beaker. So diffusion, high concentration to low concentration. We call that our concentration what? Concentration gradient. Now, this can happen naturally. No energy. What do we call that kind of diffusion? No energy. Don't need it. Just happens natural. Passive. So in your textbook, they start describing different methods of passive diffusion. So diffusion itself is a passive process. Again, we want to go from high concentration to low concentration and evenly distribute ourselves through a certain space. That's simple diffusion. Now, sometimes when we talk about the plasma membrane, we can have a little help, sort of like a little bridge. Again, this is still what kind of process? Does it take energy? It's still passive, but needs a little help. So beyond simple diffusion, we can have something called facilitated diffusion. I like the one C, because it's visually easier to understand. So there's a little protein there that will allow things to move across the bridge, back and forth with their what? Where do the molecules want to go? Concentration gradient. But remember the phospholipids that made up the plasma membrane? What did we say about those molecules? They were hydrophobic. So if somebody that doesn't mind the water and flows with the water wants to get in and out, is it going to be able to do that through the phospholipid? No, because the phospholipid's going to do what? Repel them. I'm afraid. So these little facilitated diffusion channels are going to allow small things to come in and out of the cell freely. Remember I said aquaporins? Those are proteins that will allow what? Water to do that. That's not Mickey Mouse. That's actually a water molecule. Doesn't it look like Mickey Mouse? H2O, polar, that's why it's shaped that way, and will allow it to flow in and out of the cell freely. Again, what kind of process is this? Passive, which means what? Takes no energy. We can also have little carriers, kind of like little, you know when you go on a subway, you have to kind of push through that, that little clink-clink channel? Yeah, like a little turnstile. Okay, that's what these guys kind of remind me of. Again, it doesn't take energy for this to happen, but they help carry maybe a little larger molecule in and out. So these are called carrier-mediated, facilitated because I need extra besides the phospholipids, diffusion. So little carriers. Again, what kind of process is this? Passive, no energy. Then we have the channel-mediated, which we described here, facilitated diffusion. So certain channels that will allow things to come in and out. Then they describe osmosis. The movement of water into and out of cells is so important. We give the diffusion of water its own special name. And what is the diffusion of water called? Osmosis. Yes? That's basically what it is. So osmosis is the diffusion of water. The movement of water from what? A high concentration to a low concentration. We're going to talk about the principle of tonicity and the importance of water movement. And those little aquaporins are going to help us a great deal. Now those aquaporins are going to allow water to move in and out of the cells freely. And water is going to constantly do that to make sure that the water balance is equal on either side. When I start dissolving things in my liquids, the concentration of water is going to start to go down. But again, water is always wanting to equal out in something that is a solution that has dissolved substances in it. Remember, my plasma membranes have those little aquaporins in them. So what do we say about them with respect to water? They are permeable to water. So water can very quickly move in and out of the cells to equalize the concentration of dissolved substances on either side of the cell. Some membranes will only allow certain things to come through. So if a molecule is a little bit bigger, and say it's on the outside of the cell, and my membrane doesn't allow that substance to leave, what's water going to do to equal out? Water can leave, right? So it's going to move out of the cell to equalize the concentration on either side, or into the cell to equalize the concentration on either side. So at the bottom here, we see membrane permeability to water. That might be impermeable to solutes. You're actually going to perform these experiments in the lab and see what happens to a fake cell that you make. And the cell that you make will be a semi-permeable membrane. It's tubing called dialysis tubing. So if I were to look at it with a high-powered microscope, it would have tiny little holes in it. But the holes will only be big enough to allow certain size molecules to pass through. Your membranes will only allow small molecules to pass through. Large molecules won't be able to. And you're going to see what happens to the cells with water movement that's trying to equalize the concentration on either side of the membrane. That's basically the principle of tonicity. So you see here, when I take solutes, and what's a solute? That's from the chemistry chapter. You should already know that. Yeah, that's dissolved in the solvent. In this case is water. We see at the bottom the solute is too big to fit through the holes. But remember, water isn't. So what's going to happen to the amount of water on this side of the membrane? It's going to be much, much higher than the other side. Do you see where I'm going with this? So if you mess up the concentration on one side of the other of your cells, water, which can move freely, is going to move to fix that. And that could be a problem for our cells, right? That's what the next thing in your textbook discusses when they discuss tonicity. So concentration of dissolved substances in solution and the amount of water, your osmotic balance in body, is extremely important for every single one of your cells. Remember, we're constantly, once we get to that happy point, like you are in this classroom, are in a state of equilibrium. That doesn't mean we stop moving. It means, look at the isotonic solution cell, that things are going to move into and out of the cell. And what thing are we talking about right now? Water. At an equal rate. So a couple water molecules are going to move out, a couple water molecules are going to move in. Everybody's happy. What kind of solution do I have? Isotonic. My solution has the same amount of dissolved substances in it as the solution what? Inside my cell. You with me? So what kind of solutions do you want to have in your body? Isotonic. You know when you go to the hospital, sometimes you need fluids, or sometimes you need some certain drugs to be administered to you in a fluid medium, and they hang an IV. Did anybody ever look at what the IV it contains? You have the badge, so I'm going to pick on you. You know what an IV is, right? Okay, what's in it? Besides the drugs and whatever, what else? Saline, sodium chloride. What concentration? She just took the badge off because she's afraid of it. What concentration? What's saline? Sometimes it's called Ringer solution. Sometimes it's called normal saline. What is it? Sodium chloride. Point what? 0.9% sodium chloride. Well, why the heck do you think we put 0.9% sodium chloride in our solution? Take a wild guess. Because that's the concentration of sodium chloride in the solutions that are where? Inside my cells. So say, now we call the company, we say, bring us our box of normal saline. Well, what if the guy at the factory was a little lax? Kind of lazy that day. His job is to make the concentration just right. And how does he know how to make the concentration just right? I'm going back to the chemistry chapter. Yeah, he knows all about Avogadro's number and the molar and the percent, and he knows that in order to make this solution, I need to add so many grams of sodium chloride to so many liters of water. That's his job. But he was making his solutions, and he decided, to take a little break, and then he came back, and he's like, did I add the sodium chloride to that beaker? I don't remember. I'll just add it again. Shouldn't be a problem, right? Is it going to be a problem? Yeah. Because the second solution is what we call a hypertonic solution. So now Mr. Smarty Pants added twice as much sodium chloride to the solution as he should have. What happens to the cell? Remember, water is going to do its thing. It doesn't know, it doesn't understand, it just does its thing. So if I put a hypertonic solution, a solution with more dissolved substances in it than where? Than what's inside my cells, what do we call that solution? A hypertonic solution. What's going to happen to my cells? Water is going to move out faster than it moves in. And what's going to happen to the cells? They're going to shrink. There's a fancy name for that. It's called crenation. And if I'm all shrunked up on myself, am I going to be able to do my job properly? No. If I'm kind of like dehydrated inside, am I going to have enough water for all my chemistry and all that stuff? So is that good for my cells? No. Same guy, next day, goes and takes another break, comes back, says, I can't remember, I don't want to make the same mistake again, so I'm just not going to add any. It's only 0.9%, who's going to know, right? Just send out the plain water, we're good. That's called a what? That's called a hypotonic solution. And what is that? A solution that has a much lower amount of dissolved substances in it than what? What's inside my cells. So he says that's, 0.9%. That's not even 1%. Who cares? What's going to happen to my cells? The opposite. Water's going to move in a lot faster than it moves out. And what's going to happen to my cell? Fancy name for that. Lice. Burst. Blow up. Can cells work that well when they've been blown up? No. Can you drink too much water? Yes. Now your body mechanisms are in place to help prevent too much water from building up in your system, right? So you have centers in your central nervous system that are constantly monitoring the tonicity of your fluids. When you're hypertonic, you're kind of what? You're dehydrated. So what do those things signal? Yeah, they cause your tongue to get really dry, and you go, I got to have a drink. Well, why does that happen? So that you'll do what? Go get a drink, because you need to get fluid into your body to bring your solutions back to isotonic. When you drink way too much water, you're going to visit someplace way too much for the day. Where? The ladies' or the men's room, yes? Because your renal system will get signals like, hey, a little bit too much water, let's get rid of that. So your renal system is also responsible for doing what? Regulating the tonicity of your fluids, regulating water balance. So water balance, tonicity, and the basis of tonicity is extremely important for normal cellular function. When we talk about different systems, we're going to refer to this principle. So, understand what isotonic, hypertonic, and hypotonic solutions are and how they're going to affect our cells. All of these processes that we just talked about, movement of stuff through membranes, are all what type of processes? Passive, which means what? No energy to happen. The next group of processes, not passive. What do we call them? Active. Why do we call them active? We use energy for them to happen. So active transport is going to need energy to work. And usually active transport systems are either hauling something way large into the cell or taking something way large out of the cell, or maybe bringing a whole group of things in, kind of like a moving truck, or moving molecules against their concentration gradient. That takes energy to happen. So those are some of the things that active transport mechanisms will help us do. So active transport involves some of those membrane proteins. Sometimes we refer to the membrane proteins as solute pumps, pumping in and out things that are dissolved in what? A solvent, water. There's some sort of transport system that involves energy. One very important one that we're going to talk about, especially when we talk about the nervous system and nerve impulses and the muscular system and muscle contractions, is one called the sodium-potassium pump. And what this little protein does, or ATPase, and there you see that term in there now, because what does this thing need to work? ATP, it needs energy. And enzymes are going to like catalyze reactions, right? So we refer to it as an ATPase. The sodium-potassium pump is actually going to push sodium-potassium into a more concentrated area against its concentration gradient. That takes work. So what we're going to see when we look at the plasma membrane, we are sometimes going to refer to it as a polar membrane. You know that word. What's that mean? Exactly. When we talk about the plasma membrane, we're going to see, with respect to charge associated with the membrane, when the membrane is at rest, and sometimes it'll be referred to as resting membrane potential, we'll see that there's more positive charge on the outside and more of a negative charge on the inside. And one of the reasons for that is these ATPases, or active transport pumps. Sometimes I need, and this is an example of primary active transport and secondary active transport, which they describe in your book. So, sodium-potassium pump, ATP-driven pump is going to help with the concentration of sodium-potassium inside and outside the cell. Sometimes these pumps can kind of serve double duty and move different things at the same time. And that's what they talk about when they talk about secondary active transport. The other type of thing that's going to take a lot of energy is something called vesicular transport. Vesicular transport involves the formation of a vessel or a package. Now remember, the plasma membrane is packaged with that phospholipid bilayer with all those other things in it. And we're going to see, because we have prokaryotic cells, what the heck did she just say? What's a prokaryotic cell? Thank you, honey. Did everybody sign? Prokaryotic cell. Guys, bring it back to the back. Nobody took biology? Prokaryotic, eukaryotic. Prokaryotic cells have organelles, little parts, we're going to talk about in this chapter, that are wrapped with what? Membranes. Complex, larger cells, membrane-bound things. So these cells, your cells, eukaryotic cells, that's how I remember. You have what? Eukaryotic cells are more complex and have membrane-bound organelles, little parts. And they also have the ability to make little packages within the cell for transport. So the next thing the book talks about is something called vesicular transport. Basically making a membrane-like package to transport things into and out of the cell. In order to do that, I'm going to need what? Do you ever send a box out for somebody for Christmas? You got to go get the tape, and you got to go get the box, and you got to get the paper, and you got to. Takes a lot of what? Energy to make a package. Takes the cell a lot of energy to make a package. What kind of process is this? Active. Now, bringing things into the cell and bringing things out of the cell can be done by making a package. If I were to ask a submarine who is under the water if I could board, how would I get on? Would I just open the door and swim on in? They'd get really pissed. Yes? So how do I get in to a submarine that's underwater? Nope. On a submarine, the entrance has a little sort of cordoned off place, like a little package. So you could get in with a whole bunch of water and then shut the door. And then there's little pumps that do what? Pump all the water out of that little package. And then you can open the door on the other side and get in. The cell, when it's bringing stuff in, doesn't want to just open the door and let all the other stuff from the outside come in. So it's going to kind of wrap its arms around things and create a little package to encapsulate things and bring them into the cell. There's little proteins called clathrons that hang out on the surface of the cell that are going to help initiate the formation of these little packages. These packages have a general name, and when we learn the organelles of the cell, you're going to hear a term called vacuole. Vacuole is basically any little package that the cell has the ability to make. Now, I can make packages on the inside of the cell and ship them within the cell, but I can make packages that wrap something from the outside and bring them in. That process, by the way, of making a package and wrapping it around something from the outside and bringing it in is called what? Nobody read the chapter? You don't have this all memorized yet? It's called endocytosis. endocytosis. And we can see that at the top of the diagram. So I'm creating a package, an endocytic vesicle. Basically, it's a vacuole from something that I brought from the outside into the cell. Now, if I am a cell that's responsible for getting rid of nasty little critters like bacteria through my body, do I just want to grab a bacteria, bring it into the cell, and let it run around willy-nilly? Well, no. It can cause harm. So I'm going to encase it in a package and bring it into the cell. Well, once it's in the package in the cell, well, what the heck am I going to do with it now? I'm going to kill it. I have the ability to create packages of chemicals. They're called lysing chemicals. And that vacuole that contains the lysing chemicals are called lysosomes. Why do you think I put them in a package? Because they break things apart. If I just let them run around my cell, what are they going to do? Break my cell apart. So I have to put them in a package called a lysosome. Well, sometimes I can take the two packages, the one that contains the nasty bacteria, and fuse them together, the one that contains the nasty chemicals, and create a place where I can get rid of the nasty critter, destroy it, break it apart. Now I have this big package. Sometimes it's referred to as a phagolysosome. Don't worry about it. When we talk phagocytosis, we'll see it again. What do I do with all the dead body parts? Little dead bacteria body parts. I want to get rid of them, right? Take that same package, and again, if I want to leave after I, make my visit in the submarine, I'm not just going to go open the door to leave, am I? What am I going to do? I'm going to go back into that same little chamber that we described earlier. So that package of dead little bacteria body parts is going to have to leave the cell by fusing with the existing plasma membrane and sort of opening up and spreading all those little dead body parts outside the cell. Who's going to get rid of all the dead body parts? The circulatory system is going to carry them to places like respiratory system. Who gets rid of waste? Urinary system, not the digestive system. Yeah, integumentary system. So those waste products will then be discarded by several different systems in your body, and the plasma, the fluid that surrounds your cells, is going to help transport it. What's this called when I take the package and put it out? Exocytosis. So endocytosis and exocytosis and transcytosis. And what's transcytosis? Yeah, that package traveling through the cell is what kind of process? Active process. And that's what's described in the next few pages of your textbook. Sometimes I want to bring in a lot of liquid. Like just like a little package of liquid. Sort of like, sometimes it's referred to as cell drinking. This process is called pinocytosis. And it's gathering a group of fluid using that whole package principle. So what kind of process is pinocytosis? I have to make a package. I have to go get the tape. I have to get the box. It's an active process. Yes? You with me? Endocytosis, exocytosis, pinocytosis, transcytosis. We good? Sometimes there are little proteins on the surface of cells that when they come in contact with something very specific, They cause the cell to make a package. This is called receptor-mediated endocytosis. And again, it involves very specific receptors on the surface of the cell. That's why it's called what? Receptor-mediated endocytosis. So we talk about some different proteins there that are involved with that. They talk about exocytosis. That is the opposite of endocytosis. But remember, we always have to remember that we don't want to create a hole within the cell. So there's very specific proteins, they're called snares, that are going to be involved in sort of fusing the vacuole created, that membrane-bound, whatever it is, with our membrane-bound cell so that we can release the substance through exocytosis. You still with me? So, on page 78, table 3.2, we talk about active and passive. Oh, is this just active? Yeah. Some of the different active processes. Use the tables in your book to your advantage. Has anybody noticed them? If at this point your book goes when you open it, that's a problem because you should already have worked your bindings already. See this thing? How much did you pay for it? How much? 200 what? $240. It's not a paperweight. Yes? You paid $240 for this. Use it. Open it. Read it. Yes? You honestly, your bindings should not be cracking. If they are, go open it up a few times. Because if I come and I see you like sitting someplace, I'm going to open your book to see if your bindings crack. So use it. And the tables in these chapters are going to help you so much. Because what do? They take like 20 pages of information and bring it into one place. And when we discuss the nervous system, we're going to see how important that is. We need to make the information small. So our study process should take information big. And by the time we sit down to study for an exam, we should have this much to study. Okay? And that's gonna allow you to be successful in this course. So you have to try and make the information small. You don't want to be reading this book the night before an exam. You don't even want to touch this book because it's too much. Yes? Okay. So what's this a diagram of? Key role in potassium in generating resting membrane potential. Oh yeah, what the heck was that? Didn't we talk about that earlier? What does that mean? Resting membrane potential. Our membranes are what? They're semi-permeable. But when we talk about potential, think charge. I don't know. I'm just going to blow my nose. Charge. I just said it like 3 seconds ago. What's the charge of a resting membrane? Say it louder. No. It's polar. Blow your nose again. Positive on the outside, negative on the inside. Yes? I know she just said that. How come I couldn't remember it? So that's what they're talking about in this portion of the textbook when they describe the plasma membrane and generating a membrane potential. These proteins that we've been talking about so far are the ones that are going to help move ions, and everybody knows what an ion is, right? What's an ion? It's a positive or negatively charged particle, like potassium in this picture is what? Positive. What's these big honking things with an A on them? Negative ions. So positive ions and negative ions have a name. If I'm a negatively charged ion, I am an anion. If I am a positively charged ion, I am a cation. Anybody cat lovers in the room? No cat lovers? I'm a dog lover. I love all animals, love them all. But as far as in my house, I positively hate cats. That's how I remember the difference between a cation and an anion. Sorry, cat lovers, but you gotta do what works, right? So cations and anions are what create that membrane potential. Very large proteins, negatively charged proteins. Anions are what contribute to the negative portion of the inner membrane charge. We will pick up with plasma membrane cell environment interactions on Tuesday of next week. Make sure you take your quiz by Friday on chapter what? Chapter 2.